what do we know about the function of the protein and how does it work? That's easier in Luis. Well, we know very little about the function of the protein and how it works, but what we know, it is involved in the formation of the legs and the arms of the nerve cells, the neurons called the dendrites. There are indications that it is involved in the formation of dendrites, and there are also indications that it is involved in the forming of a certain type of motor neurons called the beta motor neurons. That's the only thing that we have observed. But as more and more symptoms emerged amongst our children, we think that it is involved in so much, as I say, not directly, not by being, the protein that forms actually neurons, but by commanding commanding the function of other genes. That is the problem with ZARD, and that could explain why ZARD has such a huge variety, is because depending how functional the protein came to be, some genes were transcribed and other genes were not transcribed, and some of your proteins are working well and some of the proteins are not working well. The studies that we have done and are doing at the moment try to look at where does ZC4H2 work together with. Does it work together with DNA, with RNA, or with other proteins? And you know what? So far, it works with DNA, with RNA, and with other proteins, further complicating the understanding of ZC4H2. Now, why is this important and relevant? Because when we talk about cures for rare diseases, we know of rare diseases that affect, say, a gene that produces a protein that is a very specific enzyme, say that. And when you replace the enzyme in the body starts recovering and functioning correctly. So you rescue the defect. In ZC4H2, when you replace the protein ZC4H2 in animal models, and you take a mouse that doesn't produce ZC4H2 and you inject it with ZC4H2, or zebrafish we have done, nothing happens. It cannot be recovered, the defect. Why is that? Because ZC4H2 is involved in a lot of things. In those defects, those things already happened in an embryonic phase. Yeah. So that's one of the challenges that we see, is that when we think of genetic therapy or gene therapy, it's not as simple as taking the gene of my child, cutting it, and replacing it by a sequence that is not mutated, and my child is cured. It's not that, because again, whatever went wrong already went wrong very early in the development. So in order to get to a gene therapy, we first need to elucidate what other genes are involved, what other genes are affected by ZC4H2, and what other proteins are affected by ZC4H2. And that is a long way to go. We're not there yet.